So with that, I'm going to introduce our first speaker, Guillermo Martinez Boggio. He's a postdoc in animal and dairy sciences, mentored by Kent Weigel and Francisco Peñagaricano. He received his PhD in animal science from INRAE in France, and I'd have you come up to the podium. Let's give him a big welcome.
Thank you, Paul, for the introduction. Hello, everyone. It's my pleasure to be here today to talk about predicting methane production in dairy cows using milk spectra and genomic data. So Paul said that the first speakers will be talking about genetics, so don't stay, will be more about management than genetics. So for those that they don't know, methane, we are working on methane emissions from this big project, Greener Cattle Initiative, that is led by Dr. Francisco Peñagaricano at the Department of Animal and Dairy Science. And basically this project has the main objective to integrate different sources of information, genomics, spectra, and microbiome, to mitigate enteric methane emissions in dairy cows. The project has three main components. One of them is genetics, that I will, as I said, will not be the focus of my presentation today. What we are doing is developing a genomic evaluation for methane emissions in dairy cows. So in the future, the farmers will be able to select for cows that produce less methane. The other component is management, and is what I will be talking about today. And the third one is about microbiome. And there we are digging on different microbial communities in the gastrointestinal tract and to understand how it's related with the methane production and what are the microbial interventions that we can develop. So as I mentioned, today I will be talking about management. What we are bringing to the table is trying to predict methane production using milk spectra. So for those that they don't know, in US we have a quite unique system where farmers are collecting data at the farm, dairy herd information, and then we have more than 25 labs all across US that are sending technicians to the different farms and collecting milk samples. Those milk samples are going to dairy record processors or milk labs, state and regional ones, that are doing some processing with these milk samples. And that ends on a report. Commonly that happens once a month. So that finishes with a report that is sending back to the farmers where they can check how much protein, fat, lactose, somatic cell count each cow is having. But what I discovered was, and I learned that once I was working in France with dairy sheep, is protein, fat, lactose, those are predictions. So those are not measured directly from the milk sample that we retrieve from the farm. And those predictions are coming from where? Are coming basically from this technique, Fourier transform mid-infrared spectroscopy. To explain simply what is happening when we have the milk sample that we collect at the farm, an infrared light is going through the milk sample, and in this milk sample we have different components, chemical components. So those, when they receive the light, they absorb some energy that makes them move. And there are some other light that is not absorbed, is transmitted, and is what we can detect with this detector. So the technique basically what is getting is how much light is being absorbed by the different molecules in the spectra. And at the end, we have this result. So for the length of the mid-spectra, we have information of absorbance. So in the y-axis, you can see the absorbance. In the x-axis, the different wavelength. So you can see that we have information from the different components. So there are regions that are quantifying lactose, some others that are quantifying fat, and some others that are quantifying protein. And that is what is being used to measure how much protein, fat, lactose we have in this milk sample. What we want to do is to use this same information to predict methane production. So the extra value that we want to bring to the farmer is methane production. So in this monthly report that they receive, they have the same information plus methane production. What the farmer can do with that? A lot of things. One of them is improve their cow management. So then if they want to use a feed additive, or in the future we develop a microbial intervention, they can target a specific group of animals at their farm that are producing a lot of methane. And the beauty of this project is all the tools that we are developing on the different components, genetic, management, and microbiome, they can work together. So if the farmer wants to use only genetics, can do it. If they want to use genetics and management, they can do it too. So we are bringing tools for the farmers. In the future, if they should apply any of those to get an extra value from the market, they can do it. So today I will show the result that we have right now from this part of the project, where we are collecting methane emissions, milk spectra and genomics from 3 states, Iowa, Wisconsin, and Michigan, 7 farms, 36 trials, and right now we have 1,200 dairy cows, Holstein cows. One of the most challenging aspect that we have of this project is to collect methane emissions, and we are doing through green feed system. So it's a voluntary system that we install 1 or 2 per pen, and the cow can visit 24/7. They go there, they receive some pellets, and while they are breathing and burping inside the machine, we are collecting how much methane they are producing. And there you can see how we have installed in Arlington, for example, but also in commercial farms in Wisconsin. So what we obtain? How good are the predictions when we use this milk spectra? So what I will show is 3 different molds. So this first mold is only using productive trait, let's say daisy milk, milk, and body weight that we collect. And we are using 3 different validation approaches. So we are using farm independent. That means that for prediction, we will have a training and a testing set. In the testing set, we will use only 1 farm, let's say a farm in Iowa. And in the training set, we will have all the other farms in Wisconsin and Michigan. And we will try to predict the methane production of the cows in the farm in Iowa. Then we have another validation approach that is trial independent. So again, in the testing, we use 1 trial, could be a specific trial during the summer in Penn 30 in Arlington, and then all the other trials in the training set. And lastly, just cow independent, we create those groups at random, putting cows in different groups. So, as you can see, with this model only including daisy milk, milk, and metabolic body weight, we obtain roughly 30–40% in terms of predictive correlation. So basically what we are saying is how much we are predicting and how much we observe at the farm. What is the correlation? When we add the spectra data, so then we improve in 10–15% what we are getting, and we are on roughly 50–60% of predictive correlation, what is really good result. And the extra thing is to add the genomic data to this model, and then as you can see, there is no much different, at least for now, in those preliminary results, Because let's say that right now the data set is not as big as we expect in a couple of years, and also because they are highly correlated. So, as a take-home messages, the phenotyping is in progress. So every month we are including new cows in our data set. So that will improve our predictions in all the components of the project. And also I want to take a minute to thank all the students, visitors at the lab that are taking time to go to a farm and collect data every week during the year. That is one of the most important tasks that we have in this project. And then as I show, at least the preliminary result shows that the inclusion of spectra data, the same that we are using for protein fat, is showing promising results for methane production. So with that, I want to thank for your attention. I'm glad to take any question.
All right, thank you, Guillermo. And the floor is open for questions. Just go ahead and raise your hand if anybody has any questions. And while we're waiting for questions, I'm going to ask one, Guillermo. I was surprised that when you added your genomic component to the model, that those graphs didn't move like at all. Mm-hmm. You're saying that's just because of a number issue?
Yeah, at the beginning I was surprised too. So, but then I ran some extra analysis, and basically the explanation for that is protein and fat are traits that are moderately heritable. So those are traits that are explained, most of the variation is explained by genetics. So then when we get the spectra, basically the protein and fat is being predicted with this same spectra data. So, and then I calculate the heritability, so how much of the variation of the spectra is explained by genetics, and was around 40 to 50%. So that is why I say that is correlated. Again, we expect that with more data, we'll be able to get more difference in terms of when we include genomics or spectra data.
And you had 1,200 records so far? 1,200 cows, is that right?
Yeah, so right now we have more. The issue is, like the project, we have some farms we don't have a spectra sometimes, so that we reduce. But I mean, the objective of the project is 4,000, so I guess at the end we will have more power. Thank you.
So, Guillermo, thanks
Yes.
for the nice presentation.
Thank you.
Just a follow-up of Paul's question. You didn't show, I believe, only using genetic information and not spectra. What the result would be of that?
That was 40%. There was an intermediate between the model that I show with fixed effects or with productive traits and spectra. So at the end, when I add so we are having a very good result with spectra. So at the same time, it was hard to imagine that we will have another 10% including genomic data. You know, when they are very close in terms of how much data. But only with genomic was 40% was in the middle there. Thank you.
All right, question here.
Great talk. One question on the spectral, properties. You were showing there is a huge baseline variation there. How do you manage, taking care of those, and are those creating some huge error in your measurements?
Yes, so I think that you are asking, so she's asking about the baseline when I process the spectra data. So I mean, basically I'm using different models that account for the different peaks and the different regions that explain more about our phenotype. So there are different prior distributions that we can use to adjust the baseline and the different areas that explain more and bring more information. So we are accounting for that. But even an extra step that we are getting data from different labs and different instruments. Let's say that to measure spectra, we have different machines. So we are standardizing these two to have the same information, doesn't matter from where is coming the data. Thank you.
Okay, we do have time for one more question.
Thank you. So I have a question about the milk components, because if you think about the milk fat, some of that come from the ruminal VFA. So if you think about milk fat and you rank them based on, milk fat from high to low, and also the methane from high to low, is there any correlations in like the highest fat producing cows are also the ones that produce more or less methane? So is there any ranking you could get if we're just trying to select among a group of cows, like which one are highest methane producing cows or lowest based on their milk fat percentage?
Yeah, that is a great question. So that is something that we can do. When we work with those kind of prediction models, like a black box, doesn't matter what you put inside this model, if it works, just you don't try to understand much. But one of the point of the project is to open this black box and say, okay, what is happening here, and how that is a great point that we can check. Because at the end of the day, the connection between the spectra and the methane production is in the rumen. And then we need to dig more on this aspect. So I note and we will test it. Thank you.